Hello, everyone, and welcome. My name is Carmen Gugude, and I'm your host from Technobis today. I am happy to share today this platform with you and with our guest today, our presenter, Professor Marta Grover from Georgia Tech. She will share some of her experiences and all the information on characterization, solubility, hygroscopicity of a pharmaceutical compound, a project done in collaboration with the BMS, but you'll hear a little bit more from her soon, in a few minutes. Before we start, I would like to share a few technical details with you. The webinar is being recorded and will be made available to you after this webinar. So I think tomorrow you will receive an email with a link where the recording can be viewed again. The same for the slides. Our presenter today will make them available soon, so you will be able also to download them from our database of publications. The webinar is going to last about 30 minutes. If you have any questions related to the topic, to the case study that Marta will present today, you can directly contact her. She will also share her email address soon. And if not, you can also contact us if it's something related to our product at info@crystallizationsystems.com. We will be happy to answer your questions. Also, if you would like to share to us, we will also share everything, all the questions that they are coming with Marta. We'll try to address everything. Also, if you have questions now and you would like to make use of this platform, webinar platform, please write to us here in the question function, chat function, and we will read the questions at the end and we'll try to address all of them. Before I introduce our speaker today, I would like just in a few words to tell you who is Technobis, Technobis Crystallization Systems, for those that they do not know our company. We are a Dutch company located in the north of the Netherlands, world-leading technology provider for solid-state research, process development, formulation, and since recently with products on reaction screening. We have two product portfolios, the crystallization systems and the reaction screening portfolio. The reaction screening portfolio is a multiple reactor system with automated sampling. It is designated to address needs of chemical screening and optimization, route scouting, reaction screening, optimization, reaction profiling, kinetics, and so on. The crystallization portfolio, Crystal Breeder, Crystal 16, and the crystalline instruments. I see some names connecting today at this webinar. Some of you are familiar already with the Crystal 16 and the crystalline instruments. As you know, Crystal 16 is your instrument at 1 milliliter scale that addresses solubility, metastable zone with nucleation, induction time measurements, phase diagrams, solid state screening, you name it. This is your instrument. The crystalline instrument with the camera technology that works exactly like a microscope, or if you have a Raman spectrometer in your lab, you can connect it to the crystalline, is your instrument for optimizing or developing your crystallization processes or formulation processes. Particle size distribution is available on the crystalline with making use of an AI package software and so on. And the Crystal Breeder really for growing single crystals at microliter scale is a multiple reactor system with 32 reactors. And this is it about Technobis. And now I have the pleasure to introduce to you Professor Marta Grover. She's a professor in School of Chemical and Biomolecular Engineering at Georgia Tech and an associate chair for graduate studies. She is the advanced professor for College of Engineering and the Thomas Fanning Chair for Equity-Centered Engineering. She earned her bachelor in mechanical engineering from University of Illinois and her master and PhD, the same, in mechanical engineering from Caltech. She joined Georgia Tech back in 2003. 2011, she received the Outstanding Young Research Award from the Computing and System Technology Division of AICHE. In 2018, she received the Himmelblau Award for Innovations in Computer-Based Chemical Engineering Education. Her research program is dedicated to understanding, modeling, and engineering the self-assembly of atoms and small molecules to create larger scale structures and complex functionality. Her approach draws on process system engineering, combining modeling and experiments in applications dominated by kinetics, including crystal growth and film deposition with applications in pharmaceutical manufacture and nuclear waste immobilization. And I think these are just a few words about Marta, an impressive CV, and we are very happy to have you here today, Marta, and thankful that you are able to share a bit of science insights today with our viewers. You can try to put your slides up. Yes, perfect.
Use the pointer. Good.
Yes.
Okay, ready to go?
Perfect. Thank you very much, Marta.
Thank you, Carmen. It's my pleasure to be here today and to tell you about some of the research that we do at Georgia Tech, in particular, to tell you about this particular study on characterization, solubility, and hygroscopicity of a pharmaceutical compound. And this is a study that we completed and published last year, led by postdoc Rapunjali Prasad and PhD student Stephanie Kusevska, together with my colleague Ron Rousseau at Georgia Tech, and in collaboration with Dimitri Sklier at Bristol Myers Squibb. And in my overall research program, we're centered in process control and consider applications involving molecular organization, as Carmen explained to you in the introduction. But just wanted to set the stage to kind of show you some of the variety of applications that we consider. This is a conjugated polymer thin film that forms fibrillar morphology, paracrystalline assemblies. And here we have performed image processing to color code the fibrillar orientation of an AFM image. So we collect data, we quantitate the data, and use that for modeling and design. Here's an image of small molecule crystallization. This is actually a salt crystallization representing a nuclear waste simulant. And we also have similar looking images associated with pharmaceutical crystallization. Finally, here is a molecular simulation associated with clustering of polymers and copying information content for a project on origins of life chemistry. So these are some of the variety of applications that we consider in the group, but all of them involve process structure relationships and the kinetics of molecular assembly. And we employ a variety of modeling approaches, sometimes physically based modeling, and sometimes more data-driven modeling, machine learning, in order to quantitate our data and to make predictions for use in design. So now I'd like to get into the particular study that is the topic for today. And we were provided by BMS with this pharmaceutical compound, which was a candidate for the treatment of rheumatoid arthritis. And the compound existed as a monohydrate as provided to us by BMS. But it was observed that it can dehydrate under relatively mild sort of ambient temperature and relative humidity. And so we can see that the XRD pattern, X-ray diffraction pattern, associated with the original monohydrate form, which we're going to refer to as form 1 here. And then after being in a vacuum at ambient temperature, it would become dehydrated. And so we can see that there are some very different qualitative features associated with the dehydrated version, which we're going to refer to as form 1A. And so this structural transformation was observed to occur, and that was viewed as undesirable, that we wanted to have a crystalline form that had physical stability. So this would be important for regulatory compliance, to have a documented form and then to maintain that form, to maintain, for example, bioavailability. So also for manufacturing robustness, that we wanted to have a form that could be robustly manufactured. And so the approach that we initially took, in this study was to search for a non-aqueous solvent and thus avoid the formation of a hydrate. So that the overarching goal was to crystallize a form of physical stability, but then we were really focused on avoiding the formation of a hydrate. And so as I mentioned, we published the study this year, and if you're interested in reading the paper, you can obtain it online. It's fully open access from Organic Process Research and Development. So I'm really today going to talk you through the results from that paper. So before we got into actually crystallizing the pharmaceutical compound, we wanted to characterize the monohydrate, form 1, that we had received from BMS. And so we used dynamic scanning calorimetry together with thermal gravimetric analysis. And on the left here, we have the heat flow from the DSC. And then on the right, we have the weight loss from the TGA. But these are both measurements are being made simultaneously under a nitrogen, a dry flow. And so we start at near ambient condition, and we can see a heat flow around 60 degrees C. So it's kind of a broad feature here, but we can see that there's definitely some sort of feature. And at the same time, we can see a loss in weight. Not hugely, not large, but it's noticeable. And we were able to quantitate that at 3.8% of the mass. And it turns out that that corresponds to about 1 water molecule per molecule of the drug compound. So this is what we were expecting to see, that we had a monohydrate. So we view this as confirmation that we did have a monohydrate, and that around 60 degrees C, it would lose its water. And then we saw that we would heat it up to 200 degrees C, really no noticeable features, until we got to over 200 degrees C. And then there's a major feature which we associate with melting of the dehydrated crystal. And there's not really weight loss that's occurring, although then this we attribute to decomposition and continued weight loss above the melting temperature. And the inset here is just zooming in on this peak, but we were able to quantitate the DSC peak here to come up with an estimate of the heat of fusion. And so we actually use that later in thermodynamic modeling, and I'll talk about that later. Although if you're particularly interested in the thermodynamic modeling, you might like to read the paper, which provides more detail on those aspects. So we already had observed that the water was lost from the monohydrate when we had the crystal in vacuum at ambient temperature. We were also able to see a similar transformation that occurred when we put the monohydrate crystal into an acetonitrile slurry. And here we have the previous XRD images of Form 1 and Form 1A from the earlier slide. But here I've added the XRD pattern after slurrying in acetonitrile for 2 hours. So after 2 hours, there's not really any observable change. And after 4 hours, not so much change also, but we can see after 24 hours in the acetonitrile at 23 degrees C, we now have an XRD pattern that looks very much like Form 1A. So we were able to see that Form 1 did dehydrate and form a similar structure in the acetonitrile slurry as it did in a vacuum. So it makes sense. The acetonitrile doesn't really have water in it, so there would be a driving force, presumably, for the water to come out of the crystal. But it does take some time at 23 degrees C. So even after 4 hours, it really had not dehydrated. And finally, we performed variable temperature XRD. And this is an ambient condition, so it's not dry like we had for the DSC and TGA. And we started here with our Form 1 and then heated in the XRD. And so we have these peaks that look very similar to the Form 1 peaks until we get to 65. Although at 65, we're starting to see some transition, especially in this region over here. And then by the time we get to 75, we have a structure that looks very similar to the Form 1A. So it appears that under whatever the ambient humidity was around 65 degrees C, that is when we had this transformation, again, associated with water loss, presumably to form a structure that looks like the Form 1A structure that we saw, not that was dehydrated ambient conditions, but dry conditions. So again, these are all fairly consistent that the dehydration is occurring around 60 or 65 degrees, even in these very different environments. Having sort of established that we did have the monohydrate and that the water could be lost under a variety of conditions of temperature and humidity, we decided to do some solvent screening in our crystal 16. And so we considered 4 different non-aqueous solvents: acetone, acetonitrile, ethanol, and isopropanol. And we executed heating and cooling cycles in the crystal 16. We used the heating cycles to quantify the solubility of our crystalline form, and then cooling to explore crystallization. So here is what we saw for 3 heating and cooling cycles in each of our 4 solvents. So let's start first with ethanol. So we had three different experiments here where we used different concentration, different amounts of crystal. When we would heat, then we would hit a temperature here around 40 degrees where the crystal would disappear. So on the right axis is the percent transmission. And so when we get to 100% transmission, we take that as the point where the crystal has fully dissolved. And we use that as our solubility, the corresponding temperature. So the point where we reach 100, we can then note that time. Visually, we can come straight down vertically and see what was the temperature at that time. And then that gives us the solubility temperature associated with that concentration, which we know from the amount of material that we initially added, and that is fully dissolved. So that's how we get the solubility here. And so we determine that we're fully dissolved, continue to heat higher, hold it for some time, and then begin to cool. And what we see in the ethanol is no crystallization. So over that time frame. Now, we know that we're supersaturated because we're below the solubility once we move below this blue line. But somehow there's not enough driving force, not enough time in order to actually nucleate crystals. So it's more of a kinetic phenomenon. And if we look at the isopropanol, we see something very similar. We see that we initially are able to fully dissolve the crystal, but then we see no crystallization. Also true for acetone. We also do not see crystallization, but we do see crystallization for the acetonitrile. And we can notice that when we heat in the acetonitrile, we get behavior in each of the 3 cycles upon heating that looks quite similar. Each of these cycles looks pretty similar. We're actually, we're trying to crystallize a different form. But so on the 2nd and 3rd cycles, we probably don't have the monohydrate because there's not very much water there. The water concentration is very low. But we really don't see major differences in the solubility. So we thought that was interesting or notable. And then upon cooling, we see crystallization occurring at the 3 different concentrations at pretty similar times. You can see the pattern here of the times of crystallization are not so different, even though crystallization is a stochastic phenomenon, primary nucleation. And so, you might expect to see, not be surprised if these were at different times, but in fact, they look very similar. But we would expect to crystallize the same form in all 3 cooling cycles because in all cases, the material was fully dissolved. So I just wanted to make a few observations here. Some of those I've already made, but I might just repeat them anyway. One point that I don't think I made already is that when we're heating in the crystal 16, there may be a water loss event that occurs prior to the disappearance of the crystal that we associate with being at 100% transmission. And so we're just not able in the crystal 16 to distinguish those 2 different events. Possible they occur at the same time. We don't really know, under this sort of heating in acetonitrile how long it would take for the water loss. Recall that when we did the slurring in acetonitrile at ambient temperature, it took somewhere between 4 and 24 hours to lose the water. But now we're at elevated temperature, so it's we're just not able to say from this data when the water loss is occurring and if we have 2 steps or 1 step. But likely we have 2 steps that are occurring, and what we're observing is that final dissolution step. As I already mentioned, the dissolution in the 1st cycle, the times look pretty similar in all 3 cycles, even though we have them start with the monohydrate here. Perhaps then we have the dehydrated form 1A, that's dissolving and that we're observing here. But we really don't see differences, obvious differences in the 2nd and 3rd cycle. Observations for cooling are that crystallization was only observed in acetonitrile, although in all of the solvents, we did have supersaturation upon cooling, but probably we just didn't wait long enough in order to be able to have crystallization in the other solvents. So with the data from the crystal 16, we were able to extract the solubility and corresponding temperature and concentration. And for the thermodynamic modeling, it's more natural to use the mole fraction. And so we plotted here the mole fraction for our measured solubility as a function of temperature. And we did that for all 3 solvents. I just realized all 4 solvents, the four solvents are not actually labeled here on these slides. But the red here is the acetonitrile, and the blue is the acetone, and then the alcohols have a much higher solubility. And then we can plot that same data on a Van 't Hoff plot with the log of x versus 1 over temperature. So this is exactly the same data, but now plotted as a Van 't Hoff plot. And making some particular thermodynamic assumptions, one can derive a model in which these curves should be aligned. And so we fit a line, and actually the behavior is very consistent with being aligned. So we can also, but there's no reason that it would have to be aligned, but in fact it does have a Van 't Hoff type of curve. And then we also have ideal limit associated with having an ideal solution. And so we can calculate that from just the heat of fusion that we got from the DSC as well as the melting temperature. And then we can compare where acetone is relatively near that kind of ideal situation, but acetone is really much more favoring crystallization and phase separation. So we were able to do some additional thermodynamic modeling that you could read about more in the paper if you're interested. But as I mentioned at the beginning, our goal really was to come up with a physically stable form, something different than the original monohydrate. And so we did obtain material upon cooling in acetonitrile, and then we characterized that crystalline form. And we refer to that as form 2. So we knew from the beginning that although when we dissolve form 1, there is water in the acetonitrile, it would be a very low concentration, and therefore extraordinarily difficult to get all that water back into form another monohydrate. So that was the original rationale here was to have a non-aqueous solvent to avoid forming a hydrate crystal. But indeed, we created what we call form 2 and characterized it with XRD. You can see that the peaks are very different than the form 1 monohydrate. There's some also qualitatively different features between form 1A and form 2, although some of the features are a little bit more similar, maybe. So you can look at these and decide about what you see in these particular peaks, but there do seem to be some qualitatively different features also. This pair of peaks, for example, one peak here. So we can all scrutinize those for a while, but we observed that there were some differences in XRD, but we wanted to look more closely. And so we performed a thermal analysis of form 2 with DSC and TGA. And we were not able to really discern trend in the weight loss. So there is some upward movement actually in the apparent measured weight loss. So that was kind of curious, but we were really not able to observe obvious weight loss. Also, we understood that there really wasn't water available, though we could have acetonitrile, I guess, incorporated in the crystal that we could have. So anyway, we did not observe, any particular mass loss, but it was a little bit inconclusive due to it going up as well, I would say. With the DSC curve, similarly, we were not able to see features like, clear peaks other than the melting peak. And so that's also at a similar temperature to what we saw when we did perform the thermal analysis on form 1, that it's just above 200 degrees. And so we were able also to quantitate the heat of fusion here and got a somewhat different, but not maybe 10% different measured heat of fusion compared to the thermal analysis when that we performed on form 1. Presumably this peak there was for form 1A melting. So that's thermal analysis of form 2. And then we also measured the solubilities. And we can see here, again, we have the acetonitrile, and we have the acetone, and we have the alcohols here. So it looked pretty similar also on the Van Hoff plot. We also had straight lines associated with these different forms. Again, the acetone is sort of more near the ideal limit, whereas the acetonitrile is definitely on the side that is favoring crystallization, phase separation. And we can compare forms 1 and forms 2 here. And so we can see that because we had a different heat of fusion for form 1A and form 2, that these then in melting temperatures, these ideal limits are slightly different. But actually, that the measured solubilities for form 1 and form 2 actually are virtually the same. It's really hard to say that you could distinguish them. You could look a little bit, that maybe there's a different offset here and maybe some difference here with the alcohol or not. But I would argue that we cannot say that these forms are different based on the solubilities. But where we did see a major difference between form 1 and form 2 was on water uptake. So we performed a dynamic vapor absorption experiment. And so we vary the relative humidity from 0 to 100% and observe the mass change. And similar to what we saw for the TGA of Form 1, that there was a mass change that would occur around, there's some hysteresis here, but between, say, 20 and 40%. And the mass change was 4%, which is consistent with the DSC, consistent with having a monohydrate. However, when we performed the same experiment on Form 2, we saw that it also took up water, but it took up less water. So only about a fourth of what was taken up by Form 1. So this is actually very different. It's taking up water. Now, the original crystal that we made in the acetonitrile may not actually have had any water in it at all, or, you know, possibly it could have been a quarter hydrate, although the concentration is so low that that's kind of hard to believe. But in any case, it is taking up some water, but it's taking up significantly less water. Okay. So we do believe that these two forms are different, as evidenced by the water absorption measurement, and also that it's sort of a good thing in terms of having this Form 2 that takes up less water, takes up 4 times less water than the monohydrate. But finally, we wanted to understand the stability of Form 1. Recall that was the original motivation, was to produce a form that was physically stable. And so we can see here, if we compare the Form 2 that we had previously characterized before drying and then after drying in nitrogen, that we have really very, very similar characteristics in the XRD, that they appear to us to be the same before and after drying, and also in the variable temperature XRD. So here we start with Form 2, and this is heated under ambient condition, and we heat all the way up to 150, and we don't see any change. So we see the same structure. So we conclude that we do have a physically stable form all the way up until melting. It does take up some water. The water could be on the surface. It could be in channels in the structure, maybe that were previously held by acetonitrile. It could, but in any case, the physical structure of the pharmaceutical compound is not changing as water is coming in and out of the crystal or on and off the surface. And so we then also wanted to have slightly larger volume crystallization. We wanted to create more material and also to see about the scale and if there was a dependence on the scale. So here we crystallized in Mettler-Toledo Optimax 250 milliliters and observed with various PAT. And so originally we heated up, dissolved the material, and then we cooled down. And then we have an easy viewer and can calculate the turbidity from that. And so we make crystals, we have crystals, and then we dissolve them, and then we make crystals here. And so you can see in the easy viewer, we start off with some material, we dissolve it, and then we form crystals. And they're sort of a plate-like crystal, we don't see obvious changes in the morphology then as the crystals grow here. This also allowed us to have more material that we could use for characterization. But in all cases, we always saw the same sort of XRD pattern that we associated with Form 2. So with that, I'd like to conclude. The goal of this study was to create a physically stable form of this pharmaceutical compound. And we were able to crystallize a form from acetonitrile that maintained physical stability during variations in temperature and relative humidity. And this work was published in 2024 in Organic Process Research and Development, is available fully open access. With that, I'd like to acknowledge funding, which was provided by advanced professorship from Georgia Tech, and also facilities provided by the National Science Foundation and the material provided by Bristol Myers Squibb. And if you have any questions later, you're welcome to email me, but I'm very happy to take questions now and to discuss. Thank you.
Thank you very much, Martha. Thank you for sharing this case study. Really interesting, and once again, it shows how important are the tools that we have in our lab, and not one tool, but the multiple ones, the combination of the DSC, the TGA, the reactors, the Crystal 16, but also the Easy Viewer. So really to see what is happening, do I really see a morphology change in the form, really very nice. I have one question here, I see. Why form 2 is still the same polymorph under nitrogen if in the DVS is 0% RH humidity, it was losing the water?
So let's just go back to the DVS.
Yeah.
So we do have water that is being taken up. I also should point out here that ambient conditions, we typically associate that in a controlled internal environment 40 to 60%. And so that would be on this side where we would have water uptake. We don't always record the ambient humidity, so we don't know for every measurement, but the XRD is done at ambient conditions. So when we start, we would be on the hydrated side. Whereas the TGA and DSC are on this side. So if there was some water on there, it may have already been lost in the TGA and DSC prior to actually starting the data collection. So it took us some time to put all this together. So thank you for that good question. We needed to reconcile the difference in conditions that we would be after we did the DVS, then we realized we would be on opposite sides of the hydrated dehydrated transition. But regardless, I think what we conclude is that this is a small amount of water uptake that is not leading to a structural transition in the compound's crystalline structure. It could simply be on the surface. It might not always be 1%, depending on how much surface we have. But we're not ruling out that it could actually be stoichiometric, loosely bound water in the channels that is easily removed. And actually, this water is also easily removed, right? They're both at ambient conditions. But somehow form 2 takes up only 1/4 of the water. So and we don't see, I don't think we see really dramatic changes in the XRD between form 1A and form 2. That I think there are some differences, but at the same time they could have some structural similarities.
I think so.
But also some differences that are because of the solvent that was present during crystallization.
Yeah, indeed. And you had a confirmation from the multiple XRPDs that X-ray powder diffractions, that it was no real structural change, this in form 2. Have you, well, this is from my side, have you determined the crystal structure of these two forms?
We have not.
Okay.
We have not.
Okay. Because with that one, either from powder, either from single crystals, well, from powder, you had nice powder diffraction pattern, so could be also. And if you determine the crystal structures, you can also see if they are voids in the structure, so enough volume for a little bit of water to get in. But I do agree with you that has to be on the surface. If you look at the XRPD.
And one of the limitations in this study, we had a limited amount of material from BMS. So I would be interested to have the full crystal structure, but it really was not accessible to us.
You need to choose your battles. Another question. Somebody is thanking for the good webinar. One question: why have you chosen acetone, acetonitrile, methanol, and IPA to crystallize? Why these specific solvents?
Well, we wanted to have some a variety of solvents, polar, nonpolar. These were commonly available, I guess, not too exotic, not too toxic. I wouldn't say that these are the 4 best, but we did want to have some variation, and we had a limited amount of material, so we went ahead with these 4.
Okay. Another question. There was no particular reason why the first polymorph was a hydrate. Oh, it is a question. So is any particular reason why it was a hydrate or not? Was form 2 also good from a pharmaceutical point of view? Would have been a chosen one for further development, I think. I think that's the question.
So the original material was a hydrate, as I understand, because water was involved in the crystallization.
Yeah.
I mean, I think it would have to be, right?
Yes.
So, but this was what was provided to us as a hydrate, but it was that water was used to make it. And so we were looking for a process then that did not involve water as a solvent or a co-solvent. What was the other part of the question?
Yeah, if, well, you have a hydrate, of course, a hydrate versus a solvate is always preferred by pharmaceutical companies, of course. But looking at form 2, that it was a quarter of a hydrate maybe, or an anhydrate, was it also a good candidate for further development from a pharmaceutical point of view?
Yeah, and so I'll just point out, we could have gotten a solvate.
Okay.
And we don't know that, there wasn't any very loosely bound acetonitrile, okay. But, we really didn't see it in any of our characterizations. So, it seems that, any acetonitrile that might have been incorporated as a, as a solvate was lost under ambient conditions. Yeah. So we never observed any acetonitrile that was in the labs, but just like you could form a hydrate, you could form a solvate, of course.
Yes.
And so we didn't participate in any further studies associated with bioavailability or anything like that. So
indeed,
I can't really comment on that. I can say that form 2 does appear to also have many similarities with the dehydrated form 1A, and so. I don't know that our form would have significant differences, better or worse than, the dehydrated form.
Yeah, and to choose to take a form at the larger scale, there are many other parameters that you need to take into account, as you mentioned, also bioavailability, but also how are you going to bring it on the larger scale, what type of solvents you need to use, and so on. So it's getting more complicated. Another question. Thank you, Marta. Very interesting presentation. I have 2 questions. 2 questions from Lorena. Upright movement on TGA on slide 13. Yes. On slide 13, I believe, is it the possibility that the powder was slightly lifted in the nitrogen flow as the morphology is plate-like? Probably low density powder?
Thank you.
Could be, right? Indeed.
Yeah, no, I appreciate that suggestion.
How you prepare your sample and
we hope it's, or thinking it's some sort of artifact, experimental artifact versus a feature, but it's hard to say, but thank you for that suggestion.
Second question, have you performed slurries in different solvents for prolonged time? So like long-term type of slurries that
well, we have the 24 hours.
Oh, yeah.
So that's the longest in the acetonitrile. And we also did do some, not the time series, but we did the other solvents also, and that's in the paper.
Yeah. And another question related to these slurries, if you have performed any slurries starting from the amorphous material of this compound. Yeah.
We haven't done any studies with amorphous. I don't think we've really produced amorphous material. No.
Another question. How do you think this study can contribute to the production of the pharmaceutical compound in larger scale? Yeah, that's a complex.
Yes. So I think, solvent screening generally is, and form control is very important industrially. And so, I think the Crystal 16 was a good tool, for us to explore forms. And not that we did anything, particularly new in using the Crystal 16 to explore solvents, but it was a good tool for us to investigate these four solvents when we had limited material. And but it also provided a very nice set of data for our thermodynamic modeling. And so then we were able to fit these Van Hoff equation, and to get some insight about whether or not it was an ideal solution, or which side of the ideal limit we were on. We calculated activity coefficients. So I think that was all kind of a nice workflow that I think we could do pretty comprehensively with limited material. So many other companies also use Crystal 16 for this purpose, but we were happy with it.
Yes, thank you. Another question related to the morphological change, if you observed any morphological change between the two forms. I think you mentioned that they do not have a right?
Yeah, I mean, when you receive material, you don't know if it was milled or what treatment was done. So we really just have what's in the easy viewer images. So when we introduce the material, the form 1, this is what we saw, and we also have microscopy images, offline microscopy, but I don't have those here. But then when we crystallize, we see these plate-like crystals. But we don't know that the morphology is really different than what was crystallized originally, if this had been processed somehow. So, but this is what we have. So we do see a difference here between what we put in and what we crystallize. But I don't know that we could derive too much meaning.
Yes. Yeah, I think this is it. I think I took everything. I hope I didn't skip anything. Oh, somebody is asking on slide 9, the different color lines in the graph. Slide 9, one more. Yes. What are those? Oh, so
the different colors, the blue, red, and black.
I guess so. I think the person is not familiar with the Crystal 16 graphs then.
Okay. So the black one in the acetonitrile was 6.6 milligrams per gram of solvent, and then the red is 13.0, and the blue is 25.4. So we just had different amounts of material that were added. And so, this is basically 16, the 16, Crystal 16, we have 4 different solvents. And then, well, here we have 3 different compositions, so 12, I guess. So we use that too. So this is, one run. It took 30 hours, but, it would run automatically and continuously to generate all of this data at one time.
Okay. I think this is it. Once again, thank you very much, Marta, for sharing with us this case study. It's really a nice case study, interesting, maybe a problematic if you want to bring it on the on a larger scale for BMS, but definitely a very nice case to work on. If we receive any other questions in the meantime, we will share them with you, and when you have some time to answer them, or you might receive directly. Thank you very much. Thank you all for joining us today. You will receive a link tomorrow with the recording where it can be found. Usually it's on our website under the webinars. Thank you and see you soon.
Thank you.
Thank you.